All right, welcome back, everybody. Hope you enjoyed your Wednesday and took a look at that beginning of our kidney stuff. Before we move on to our renal system from top to bottom, we're going to finish up talking about our acid-base homeostasis. So how we balance the pH of our blood. Okay. So quick overview of where we were on Monday, right? Okay, so we were talking about basically what we expect to see in our arterial system when we're looking at pH. Okay, so where you would be measuring would be arterial blood, and you'd be looking at the pH to figure out if everything's going okay. And we've got two types of problems, right? So we could have a pH that's too low, right? So if you have a pH that's lower than 7.35, we're going to call that acidosis. Okay. And on the other side, if we've got a pH that's higher than 7.45, oops, lower than 3.5, higher. There we go, higher than 7.45. We've got alkalosis. No, I did put the symbol on the wrong side. Wow, my math symbols are not great today. Sorry, gang. Okay, our pH is higher. Okay. There we go. Too high. Okay, cool. So in this situation, we've got a couple different reasons. So if our problem is that our CO2 is too high, right? CO2 is acting as a biological acid, right? This means your problem is the lungs. So we're going to call this respiratory acidosis. And if on the other side, the reason for our pH being above 7.45 is our CO2 being too low, right? That's also a respiratory problem. So that's respiratory alkalosis. So that's all the bits coming from our lungs. But we have other contributions to pH, right? So if we're seeing this pH problem for any other reason, still going to be acidosis or alkalosis, but now we're going to call it metabolic. And as you can see, I'm trying to start us going through that chart that we're going to see at the end of the lecture. Okay, so this is how we're looking at things to start, right? And we know that we've got ways to fix pH. So the first thing we can do if we have a pH disturbance is we can try to buffer it out. And our main buffer is bicarbonate. Our second way of fixing pH disturbances, which would be an actual fix of the pH, is going to be respiratory compensation. So changing your breathing's pretty easy, assuming that this isn't a lung problem. Okay, so we're going to have respiratory compensation. So that's our lungs fixing stuff. And then our third way of fixing pH is going to be renal compensation, which is where we had just about gotten to on Monday. Okay, so this is where our kidneys are going to fix our problem. Okay, that's kind of what we did on Monday in brief. So we're going to clear that out and we're going to jump back into your actual slides. All right, so we left off here, okay, thinking about our disturbances. We've been classifying disturbances to acidic, acidosis, low pH, to alkaline, to basic, to high pH. And we had moved from trying to figure out, is this a respiratory problem? or is this some kind of metabolic problem, to starting to think about compensation, right? So first thing we can do is just buffer. That's not actually fixing the problem. That's just kind of soaking up extra hydrogen ions, extra acids. But with respiratory compensation, in a couple of minutes by varying our breathing, we can actually regulate pH, actually change it, because we can change the amount of CO2 in our bloodstream. Okay? So if we want to get rid of acid in our blood, right? One thing we can do is decrease our CO2 levels, because CO2 is acting as an acid. Right? So to do that, you'd breathe faster because you want to breathe out more carbon dioxide. So that's one way we can regulate our pH, by varying our ventilation. But we can vary a pH that's either too low or too high with ventilation changes. So if we've got the opposite problem, right, we don't have enough hydrogen ions in our blood, we're a little too basic, we can build up CO2 levels in our bloodstream as well. So to do that, we just breathe a bit slower. So decreased ventilation would take us in the opposite direction. Okay. So here we got that in flowchart form. So here we're looking at a case of acidosis, so a low pH, which is the same thing as saying high acidity, right? So you and I have to be careful about our directions of our arrows as we're going through these things, because we've got a lot of trade-offs, right? But essentially, we're going to detect this change in pH with our peripheral chemoreceptors when we sense this problem. One of the first ways your body's going to try to compensate, try to fix this, so we're going to increase ventilation to get rid of that excess acidity by breathing out carbon dioxide. So that should hopefully bring your plasma concentrations of carbon dioxide down, so we can see our partial pressure of carbon dioxide in the blood falling here in response to that increased ventilation. And that should be bringing that plasma pH back up. Okay. Once we're back to normal in that 7.35 to 7.45 range, this should stop. Those chemoreceptors are going to stop sensing a problem. Okay. So that's the lung piece. Now we're going to jump into the kidney part, or renal compensation. Okay. So I'm going to ask you to predict which of these movements of our bicarbonate ions is going to be able to return our plasma pH to our normal range, that 7.35 to 7.45, if we have respiratory acidosis. So we're thinking something's wrong with the lungs. This is something going on with our breathing causing this excess acidity. Okay. So we might want to change this using something other than our lungs. Okay, let's take a look. Okay, cool. All right, so what we're thinking about here, right? Let's think about the two kind of spaces we're working with as we think about how we're moving our bicarbonate ions. We'll look at different cells and some pathways specifically in a second, but fundamentally we're thinking about our bloodstream, right? And we're thinking about our future urine going through the kidneys, going through the nephron, right? So we'll just write nephron there, I guess. Okay. All right. So when we're thinking about how we're moving our bicarbonate ions, right? If you're reabsorbing stuff, what we're saying is we're moving stuff into the bloodstream, back into the body, right? So this would be moving bicarbonate ions into the blood, right? Which means we have more buffer now in our bloodstream. So this can soak up couple more hydrogen ions that can soak up extra acids, act as more buffer in the bloodstream. So this is why this would fix our pH problem coming from excess acidity due to malfunction of our respiratory system, right? So for the other two, right, we're just looking at bicarbonate ions basically being peed out here, okay, being excreted, or secretion would be moving from the bloodstream, moving even more of our bicarbonate ions here into the nephron and then subsequently out of your body. Right, so both of these would be removing buffer ultimately from your bloodstream. So this would make the problem worse for those other two. So that's why we want the reabsorption. Okay. And that's where we're going. Okay. So when we're looking at renal compensation, this is slower. This takes hours to potentially days to fix a pH disturbance. But it's important that we have this mechanism, especially because we might have a problem with our lungs, right? So what we're going to be regulating when we're looking at the kidneys and we're looking at this renal compensation is we're going to be doing a little bit of regulation of hydrogen ions themselves, specifically their excretion, and we'll see that we'll be secreting some hydrogen ions. But we're going to be focusing in a lot on bicarbonate. We're going to be changing the bicarbonate levels in the urine, but especially we're interested in what are we doing to the levels of bicarbonate in your bloodstream, because bicarbonate is that really important buffer. Okay. So your kidneys regulate excretion of hydrogen ions and bicarbonate, and they regulate things like the synthesis of bicarbonate in the renal tubules, which we'll take a look at how that's different in a second. Okay. So when we're looking at cases of increased acidity, which we often are, right? So we're thinking increased acidity, going to be buildup of hydrogen ions in the blood. Maybe we're seeing buildup of carbon dioxide in the blood, right? We want to get rid of this effect, right? One of the things that we're going to be able to do with our kidneys in that case, right, just like our drawing. So we got blood here. We've got our kidneys and our future urine here, right? So if you want to remove acidity, secretion of hydrogen ions takes them out of circulation. So secretion will be moving it into the nephron and then out, eventually excreted out of your body, right? So that's our secretion, okay? Secretion, there we go. Okay. And then these other two bits have to do with our bicarbonate, and they're both going to pull bicarbonate into the body, right? So we might be pulling it from the fluid that's in the nephron. So that would be our reabsorption of our bicarbonate. Okay. And the other thing we can do is in the cells of parts of the nephron, in these two cells lining our tubule, we can make bicarbonate ions, and then those would move into the bloodstream as well. So this is our synthesis piece of our bicarbonate, right? So these are sort of three different ways that we can get rid of this excess acidity. We can actually just push it into your urine, or we can make more of this buffer or pull more of the buffer back into your bloodstream to account for it. Okay. Different parts of this are going to be happening in different parts of the nephron. So different parts of that ductwork, that network of tubules inside the kidney. Okay. So we're going to see that in our proximal tubule, right? So we're going to draw a real quick little nephron here. Okay. So beginning of our nephron, we've got a glomerulus. Proximal part, proximal always means the first part, the nearest part. All right, then we got a loop. All right. And we got a distal part. Distal always means farther, just like it does in the anatomy lab. And then we have a collecting duct where multiple tubules eventually come together. Okay. So I'm just gonna put some little stars for where we're talking about right here, because I think that helps me anyway. All right, so in the proximal tubule, we're talking about this first part, all right? In that proximal tubule, we're going to see a lot of the reabsorption of bicarbonate happening as we go through the parts of the nephron and what happens in them overall. We're in general going to see that the proximal tubule just reabsorbs a lot of stuff. True for bicarbonate the way it will be true about things like water and glucose and all that jazz later. Okay. So we're going to have some bicarbonate ending up in the nephron, and then a lot of it we're going to just immediately reabsorb back into the bloodstream because we don't want to lose buffer every time we filter some blood. At the same time, there will be a little bit of hydrogen ion secretion happening, so moving hydrogen ions, extra ones, into your urine. Okay. Usually about 80 to 90% of our bicarbonate is immediately getting pulled back into your body. You can see sodium is happening there too, while we're getting rid of some hydrogen ions. Okay, so that's our proximal tubule. And then in our distal tubule and our collecting duct, so that's like here, and let's say here, right? Okay, so at the end part, basically, at this point, we've already reabsorbed up to 90% of our bicarbonate. So there's not a lot left that we could be reabsorbing by the time we get to the end of this system. So we're going to see that at these points, as we're heading on our way out of the kidney and out of the body, right before we leave, this is where we might be making some new bicarbonate ions, right? So we might be doing bicarbonate synthesis here, right? The direction respective to the blood would be the same. So we'd be making new bicarbonate ions and then just immediately pulling those into the blood. The only difference here is that we're not pulling preformed bicarbonate ions from the filtrate that's in our nephron. This is kind of brand new. Okay. Hydrogen ions here can be secreted as well, just the same as we see in the proximal tubule. Okay. So two different kind of mechanisms, but in terms of directions, we're thinking about the same thing: bicarbonate into the blood, hydrogen into the urine, out of the blood. Okay. So here we're looking at that first location first, right? So we're looking at the proximal tubule in an epithelial cell lining that nephron. Okay. So the point here we're looking for, all right, we want to understand how we are reabsorbing our bicarbonate ions. Okay. So HCO3 minus is our bicarbonate. Okay. And we are going to be secreting hydrogen ions. Okay. So the secretion part, we might be able to see immediately. Right, so remember on these diagrams from your book, when you see a red arrow, that's active transport, so some kind of pump. When you see green, that means that's in a favorable direction. So you'll see it sometimes as one of the two things in secondary active transport. It just might be purely passive. Right? But when we're looking at our hydrogen ions, right, we can see that we're just actively pumping hydrogen out of the cells and into our tubular fluid, right? So that tubular fluid eventually becomes urine, right? So this is going out of the body. Okay. So that's one piece, that secretion of hydrogen ions, removing excess acidity here. It's the reabsorption of bicarbonate that's a little more complicated, okay? But fundamentally, what we really want to be focusing on, right, is where that bicarbonate is going, which is back to the blood. The blood will be over here. Not pictured, but that's what's on the other side of the paratubular fluid. Okay. So to do this, to reabsorb it, we're going to see we've got lots of solutes in this filtered fluid that's going through these early parts of the nephron, through the proximal tubule, including some bicarbonate ions, right? So some bicarbonate ions are in the fluid that enters the nephron, okay? And in order to put those back into your bloodstream, we're going to convert them into something that has a really easy time getting into cells. So we're going to take those bicarbonate ions and we're actually going to convert them into CO2. Okay. So to do this, we're going to take some of the hydrogen ions that are in this tubular fluid. Some of those are coming from this secretion of hydrogen ions. We combine them with bicarbonate ions, turn that into carbonic acid here, right? And then when we make that carbonic acid, we got an enzyme that can break it down into water and CO2. Right, so this is our carbonic anhydrase, that enzyme that's on this apical membrane of our proximal tubule epithelial cell. Right, so our goal was to turn this bicarbonate ion into something that can just diffuse across this cell membrane easily. So we did that, turned it into some carbon dioxide. We had to put some hydrogen in, but now we see that carbon dioxide comes into the cell. Once we have it in the cell, we can put it back into the form we want to pull into the blood. So you can see we are basically just reversing that reaction. Right, so now we're taking that CO2, adding more water in. We got water everywhere in our body. Here we can see that carbonic anhydrase, that same enzyme. We've got some floating around in this cell, turned it into carbonic acid, which then dissociates back into those two ions. Right, so now we have our bicarbonate back inside this cell. We also have that extra hydrogen ion. That's fine. We already have this pump. We already mentioned we got pumped to secrete out those hydrogen ions. So that just cycles back out. And we take this bicarbonate ion, we pull it into our bloodstream. We might be pulling it along with some sodium, or we might be doing countertransport with some chloride ions. So there's a bit of an interplay here with how your proximal tubule is handling other ions, other electrolytes. But big picture, we took the bicarbonate in this fluid, converted it to carbon dioxide, inside the cell, convert it back to bicarbonate, and move that into the blood. So that's this reabsorption piece. Lots of steps, but fundamentally, just moving it from one place to the other. Okay. Now we're looking at that second location where we adjust our bicarbonate levels, right? So we're looking at the distal tubule now, right? So that distal tubule and collecting duct specifically in a type of cell called the intercalated cells, right? Later on, right? So this is after we've done a lot of reabsorbing, a lot of adjusting of concentrations of all kinds of things in our tubular fluid, right? That tubular fluid is almost ready to be urine, right? So this is still going to go out of the body, right? And our blood is still over here, so we can remember our directions. Okay. So here, we're going to be focused on, again, doing some secretion. Okay. So we're still going to be secreting some hydrogen ions. So nothing about that is really different than when we were looking at our proximal tubule. You can see we just, again, have a pump for secreting hydrogen ions. Right? Okay, so we could do that for our secreting hydrogen ions. But the other thing that we're doing that is a little bit special here is our synthesis of bicarbonate. Okay. Now, I find this difference a little bit subtle. So what I want you to focus in on is first just look at this tubular fluid, right? Looking at the contents of the tubular fluid. You can see some things in it, but none of those are bicarbonate ions, right? So if we were looking for a bicarbonate ion in the tubular fluid, right? We can have lots of question marks here, right? We don't find any, right? That's not something that's labeled here in the tubular fluid because we soaked most of it back up, right? We reabsorbed it already in the early parts of the nephron and the proximal tubule, right? So we don't really have any more to reabsorb. So we're synthesizing instead. The way that works is using a bunch of those same pathways for reabsorption. It's just we're not starting with bicarbonate ions in the tubular fluid. We're kind of starting in the middle of that process that we looked at on the previous slide. So we're actually just going to start with some carbon dioxide in the cells of the distal tubule and collecting duct. That carbon dioxide can come from a lot of places. It's a gas, it's just around, but we also know it's one of our waste products from metabolism. So these cells are making CO2 all the time as they make ATP. So CO2 is not in short supply in any of our cells. So we can take some of that CO2, combine it with some water, use our handy dandy enzyme, our carbonic anhydrase, make our carbonic acid, and then that'll split up and we have our bicarbonate ions. So these ones count as new bicarbonate ions that we synthesized because we didn't get them out of the tubular fluid, right? They originated just as carbon dioxide in the first place. Okay? And that's really the only difference here that we're going to focus in on, the fact that this bicarbonate ion is new. We're then going to take it and move it into our bloodstream. All right, so you can see here that this is happening along with some counter transport of chloride ions. We're not really focusing in on those. So if you were dialing in on exactly what types of transporters you see in these cells, if you go on to do research on the renal system, you care about that difference. But for our purposes, we're interested in the direction. Right? So we got this new bicarbonate moving into the blood. It can then act as a more effective buffer for our pH. Okay. Finally, there is one kind of last-ditch effort for adjusting really severe acidosis, for compensating for really severe acidosis. You'll see one final mechanism in our renal system. And that's using something called glutamine. So glutamine is one of our amino acids. And we can turn glutamine into bicarbonate ions as well, right? So here you can just see some glutamine. This process is different than that constant conversion between bicarbonate ions, hydrogen ions, carbon dioxide, and carbonic acid that we see over and over again. So this pathway would look a little different. But fundamentally, in really severe acidosis, we might be able to do a little bit of synthesis of bicarbonate even in the proximal tubule. So this is in that earliest parts of the nephron. If none of our normal compensatory mechanisms are working, this might kick in and you might start breaking down this amino acid to get yourself some more bicarbonate, which you would then pull into the bloodstream, same as usual. The byproduct of this would be a buildup here of ammonium in your tubular fluid and then in your urine, because that's kind of a byproduct of how we break apart the glutamine here. So you can see that we converted our ammonia into ammonium here on the other side as we were making this new bicarbonate. So hopefully we don't have to use this mechanism, but it is there. Okay. So dialing back again, more ways of thinking about this, right? So we have first our acid-base disturbance in the first place. So we have our Henderson-Hasselbalch equation that tells us how to calculate pH. We know that for our purposes, our expected pH is going to come out to about 7.4. We got a little wiggle room on either side. And if we do the math, that's going to give us a stable ratio of bicarbonate ions to carbon dioxide that we're looking for in our bloodstream. So that ratio is 20:1. Okay. So when this ratio is off, that is going to result in a pH that is off, either too low, acidosis, too high, alkalosis. Okay. So if we have too few bicarbonate ions for the amount of carbon dioxide we have, acidosis, low pH. If we have lots of bicarbonate ions and not so much CO2, your pH is going to be too high, alkalosis. And the kidneys are really primarily regulating that bicarbonate, either just through reabsorption or through synthesizing new bicarbonate. And our lungs can regulate the other side of that ratio, the CO2 part. Okay. So when we are looking at what's going on with a patient big picture, like say we have someone who's in a state of respiratory acidosis. So we would notice that that pH is too low. It's below 7.35. And reason for this in respiratory acidosis is probably hypoventilation, right? We're breathing really slowly, which means carbon dioxide is building up in the bloodstream. It's not having enough time to breathe it out, right? We don't have enough breaths per minute. So we got this buildup of carbon dioxide in the blood, which is eventually going to lead to a buildup of hydrogen ions in the blood. Okay. So if you're hypoventilating, you're probably not doing it on purpose, right? So it's probably going to be hard to fix using the respiratory system because it's happening for a reason. So our way to fix it, our way to compensate for it, is we see renal compensation. So renal system is going to try to make the math work. It's going to try to keep that bicarbonate to carbon dioxide ratio at 20:1 by adjusting our bicarbonate ions especially, right? So that bicarbonate reabsorption is going to be the easiest piece. We're going to see lots of hydrogen ion secretion happening along the same time. We might also even sometimes synthesize some bicarbonate, but that would go back in the bloodstream, okay? In cases of respiratory alkalosis, right? So in this case, we're going to see really low carbon dioxide levels because you're hyperventilating. You're breathing super, super fast. You're breathing out way more CO2 than you would usually. So you're seeing lower and lower levels of hydrogen ions, lower and lower levels of acidity. So the pH is going up and up. Again, probably not hyperventilating on purpose. So to fix your breathing, that would be great. This problem would go away. But while you're still hyperventilating, we're going to try to compensate with our renal system. So you're going to go into renal compensation. And in this case, we're going to try to hold on to any acidity in our blood that we can, right? So we're going to try to slow down the rate at which we're secreting hydrogen ions. So we don't want to be taking them from the blood into our proximal tubules, secreting them and then peeing them out, excreting them. We want them to stick around in the body. So we're going to slow that down. And we're also going to slow down how much bicarbonate is coming back into the bloodstream. If you don't have much acidity, you don't want a lot of buffer in the bloodstream either. So we're going to decrease how much bicarbonate we reabsorb. Okay. Now, we hopefully remember that respiratory problems aren't the only problems. We have metabolic reasons for both acidosis and alkalosis as well, like dietary reasons or renal disease. There are a couple of different things that we can kind of classify in this metabolic acidosis and alkalosis range, right? Just anything that's not CO2. So thinking about the metabolic acidosis, maybe this has to do with your breaking down lots of proteins. Maybe you've got a lot of lactic acid buildup, something like that. But we got an increase in hydrogen ion concentration and acidity. This is just not because of carbon dioxide. It's some other reason, okay? And so depending on the source of that, you could have respiratory and renal compensation if it's like a dietary cause. But we might have a renal cause for this. You might be in metabolic acidosis because your kidneys are failing, right? They aren't working properly, in which case you're really just going to do respiratory compensation. You're just going to fix this with your lungs, okay? And regardless, respiratory compensation is faster, happens first, okay? So we're going to start breathing faster, right? We're going to try to get rid of this excess acidity by lowering our CO2 levels so that our ratio of bicarbonate to CO2 kind of fixes this a little bit, right? And if it's not a kidney problem, we'll also do renal compensation a little later if this doesn't fix the problem. So in this case, we're going to increase how many hydrogen ions you're eliminating as waste. So you're going to increase how many hydrogen ions get secreted into the nephrons and then out of the body as urine. We're going to increase how much buffer is coming into the bloodstream. So we're going to increase the bicarbonate we're reabsorbing, and we're going to be increasing our synthesis of new bicarbonate, potentially even in our paroxysmal tubules, right? Finally, metabolic alkalosis. So we're seeing that we have really high pH for some reason that doesn't have anything to do with your CO2 levels, okay? So either your respiratory or your renal systems could fix this unless it's a renal problem, in which case it has to be just the respiratory system. Respiratory system is faster. So first thing we're going to see is we're going to slow down our breathing, let some CO2 build up to try to balance out that pH change. And then after some time, if this doesn't fix it, and we don't have a kidney problem that prevents it, you'd see renal compensation potentially kick in which case we're going to slow down our hydrogen ion secretion. We're going to slow down how much bicarbonate we're reabsorbing, and we're not going to make much new bicarbonate because we don't want it in our bloodstream in this situation anyway, right? That would raise the pH, not useful to us. Okay. So that takes us to this big chart of how we can assess renal disturbances and compensation by doing a blood sample, right? So what we start with, right, is we're going to look at the pH in the blood that we're drawing, right? It would have to be arterial blood in this case, but if you sampled some arterial blood, right, you start by seeing, is this pH out of range, right? So if our pH is too low, acidosis. If it's too high, alkalosis, right? And then the question that I find to be easiest next is to figure out, is this a respiratory problem or not, right? So is this your breathing or is this something else, right? So first thing we'll check is those partial pressures of carbon dioxide, right? So in the case of acidosis, if we see a partial pressure of carbon dioxide, it's above where it should be. And this is where you want to remember a reference number, right? So remember that your partial pressure of carbon dioxide in systemic arteries should be 40, okay? So if you see a partial pressure of carbon dioxide that's higher than that, right? That means this is a respiration problem. So this is respiratory acidosis, right? If you see that same low pH and it's not because you have a high level of carbon dioxide, probably what you're going to see is that your bicarbonate is a little bit low. Not going to require you to remember this number, but our reference point for our expected levels of bicarbonate is 24 here. Okay. So in this case, we don't have enough bicarbonate to balance out all our acids. Okay. So that's going to be something that we would see in cases of metabolic acidosis. And because we're looking at bicarbonate levels here, right, this is probably going to be a problem with our renal system if our bicarbonate levels are off, right? This isn't measuring like lactic acid or something. It's specifically about bicarbonate, which is the job of your kidneys, right? So in that case, we got a bicarbonate issue creating that low pH. You're going to compensate with the respiratory system. Versus if you're in respiratory acidosis, you compensate with the renal system. You always just compensate with the opposite one, basically. Okay. Now that is then going to have an effect on other values in your blood. So if you had a really high level of carbon dioxide in your bloodstream causing that acidosis, causing that low pH, and you do renal compensation, you're going to see that that's going to pull your bicarbonate levels to higher than normal, right? because they're compensating for something, right? They're making up for the fact that these CO2 levels are high, right? They're adjusting the bicarbonate. So we're adjusting that 20 bicarbonate to 1 carbon dioxide ratio, and your pH will go back to normal eventually. But you didn't change your CO2 level with a kidney here. Okay, so same logic when we're looking at the metabolic acidosis, right? You can see we're compensating for this low bicarbonate by lowering our carbon dioxide levels so that you don't have as much carbon dioxide to buffer out. So it doesn't matter so much that you don't have enough buffer. Okay. Now let's do a practice question. You can keep this chart up for yourselves, but we're gonna have our example patient here. So we're gonna have a patient who's unresponsive, blue lips, you're taking a blood sample, say it's arterial blood. You're gonna notice a pH of 6.9, partial pressure of CO2 of 45, and high bicarbonate levels. What do we see going on here? Okay, great. Yeah. So that's how you'd read the chart essentially. So you're starting by reading our pH. 6.9 is definitely too low, right? So that's acidosis, right? And 45 is too high for our partial pressure of carbon dioxide. So this is a respiratory problem. Okay. So if it's a respiratory problem, respiratory acidosis, probably already know that we're going to have renal compensation to adjust for it. And to confirm that, we can see that we have high levels of bicarbonate ions in the blood, right? Because the kidneys are pulling in extra bicarbonate, right? So we're reabsorbing more bicarbonate than usual primarily. Maybe we even synthesize some new bicarbonate in cases of extreme acidosis. So I recommend practicing with that chart and kind of walking through these steps on your own. I find writing my own flow charts is a helpful way to kind of get your mind around these. Okay. Questions about our pH balance before we go back to the beginning with the kidneys? Okay. We'll have a quick peek at our next topic. Okay. So we're going to be running through the parts of the nephron. So in your activity for Wednesday, you looked at our kind of big bird's eye view of the structural parts of the nephron. You looked at the blood flow to the kidneys, and you looked at micturition. So you looked at that process of forming urine and expelling it. Now we're going to dive in more detail on each of the parts of the nephron and actually how are we filtering stuff? How are we secreting stuff? Kind of like we just did for bicarbonate. So we're going to start by overviewing, right? We've got a lot of functions of our urinary system. So we're going to be really interested in water balance eventually, but we're going to be in general regulating, I'm not going to say everything in your body with your kidneys, but a lot, right? So we're going to regulate the composition of ions in the plasma using the kidneys. So we're going to affect our sodium levels. We're going to affect our potassium levels when we're thinking about that ionic composition. We're going to regulate plasma volume. So that's like how much blood you have in your body. And that's really where the water piece is going to come in. If you have more water in your blood, Functionally, that's more plasma volume, right? So that's going to link us back to this relationship actually with mean arterial pressure and blood pressure, right? Your kidneys play a role in that because they play a role in actually how much blood are you moving through your system. They also regulate osmolarity. So they're going to regulate where does water actually want to move relative to the blood, relative to the plasma. We've just taken a look at that regulation of pH. Your urinary system is also responsible for making sure your body's getting rid of the stuff it wants to get rid of. So as stuff gets filtered through your kidneys, you're gonna be removing a lot of waste products and foreign substances that are in your bloodstream and sending them onto your bladder and out of your body. We're also going to see kind of at the end that we have some hormonal functions of our kidneys as well. And they do some activating of vitamins as well as making some new glucose for us. So a couple of topics that we have seen a little glimpse of before, right? Our erythropoietin, we might remember, was one of our signals for making new red blood cells as an example. Okay. So our first step of normal function kidney is we're going to have blood coming into the kidney, right? And it's gonna come in, getting smaller and smaller branches of our bloodstream until we get to arterioles. And we're gonna have an arteriole going in and an arteriole going out of this structure called the glomerulus, right? So we can see that here, right? Blood coming in through an afferent arteriole. We got a bunch of stuff looping around. That's your glomerulus. It's a capillary bed. And then the efferent arteriole going out. Okay. So this is where filtration is going to happen. So everything in your plasma, everything in your bloodstream, except for proteins, basically, is going to end up in the nephron in that filtrate to start with. You don't want to pee it all out. You don't want it to all become urine, but it is all going to go into the nephron. And then your nephron is going to decide what gets to stay in your bloodstream and what doesn't. Okay. So in terms of the amount, you have about 125 milliliters of filtrate per minute or 180 liters per day of protein-free plasma going into the glomerulus and then going into your nephron. So this is why reabsorption is super important. You could not lose 180 liters of fluid per day as urine. You would die like basically immediately, right? So this is why kidney function is super important. You absolutely cannot lose that much fluid. You don't have that much fluid. Okay. So that fluid, this filtration step, is crossing a membrane. So when we're looking at that blood, and you can't see it because it's covered by this yellow stuff or beige stuff, that beige stuff is a special type of cell that we see in this barrier in the glomerular membrane. Okay. So we got a special type of capillary first. Our fenestrated capillary, you can see it has extra big holes in it here. We got a basement membrane, that's just glue. And then we have this capsule layer made of a type of cell called a podocyte. That's for foot. It's got these foot processes called pedicels with filtration slits between. Functionally, everything except proteins can get through this kind of filter. So we'll pick up there later. You might want to take a look back at your net filtration pressure stuff, because the thing we'll be starting with on Monday is what the pressures look like, what the Starling forces look like for filtration in the kidneys. And it's just going to be essentially some minor tweaks on that previous topic.